So I'm very pleased to see so many people here because I guarantee it's going to be a fantastic seminar. So I'm delighted to introduce Dr. Jesus Valenzuela from NIH. Jesus is one of the leading figures in the role of vector saliva and from composition to function of all the molecules, and more recently into a translational component looking into vaccines against the pathogens that these vectors transmit to humans, and also to the discovery of many of the salivary markers that we use in the field to validate our vector control interventions. Jesus did a PhD. He's originally from Mexico, and did his PhD at the University of Arizona with Jose Rivero, who's been the guru of xylons, but then passed that to Jesus more recently. When Jose Rivero moved to NIH, I think in the 90s some point, Jesus was a postdoc in Jose's lab and carried on with all this investigation using ticks, mosquitoes, and more recently, I would say probably the last 15 years, more into sandflies, saliva, rolls, composition, function, etc. So thanks for coming over, and delighted to have you here.
Thank you, Alvaro. Thank you for the nice introduction, and hopefully it will be a good talk. Well, I'm delighted to be here, and please, it's going to be a talk of different topics. You are welcome to interrupt me in the middle. And good news, I have only 30 slides or less, meaning that I don't saturate talks. I'm famous because I give short talks. I prefer time to talk to people and to get feedback from the audience. And actually, many of the major discoveries in the lab, or two of the major discoveries in the lab, came from students that gave me an idea. And I always acknowledge that. Then if you give me an idea today, I'll be very happy. Then this is the same fly. This is the insect that I'm working with. This is the vector of leishmaniasis. I did something different today because instead of putting the word saliva, we're learning that saliva is extremely important for blood feeding to prevent, yes, water, I'm thirsty. I think I don't know if it's the humidity of too much beer. I don't drink that much beer until I came here, my God. All right, kidding. What we're learning now, I'm going to show you today, is the impact of saliva, of vector saliva in transmission. But I'm going to add two components. I'm not going to show you a lot of data about the other components. I'm just going to give because Sharon Kamhawi, a member from the lab, is coming in September, and I'm going to give you the other side of the story. But the theme of the lab right now is this. And for me, it's good to communicate this. It's not only saliva. We're calling this vector-derived factors. Every time that a sandfly bites, it's not only the parasite, it's other factors coming from the vector. One of these factors is saliva. The others are exosomes, PSG, from the parasite, and I'll show you other data later. But that's how the importance of this then. And also I'm going to emphasize the importance of the skin, which actually I'm going to start with that. Every time, if you check the literature, and you go right now to any vector-borne disease, right now, and try to tell me, and actually if you find it, let me know, because I've been looking for it. And I'm gonna give you an example of sandflies. Every time that the sandfly takes a blood meal and is infected, it delivers parasites, okay, into the skin. Vectors that transmit into the skin. And together with this injects other components like saliva. Well, I'm gonna leave this to the side. But we know a lot about what's happening later, weeks or months later. We know about the disease. We know probably what drugs. We know many aspects of the disease later on. We know a lot now about how the vector gets infected, how go from whatever stage to metacyclics that makes this vector more infectious. But the initiation of the disease, the initiation of what happens after the bite, I'm talking about minutes, hours, is unknown. It's incredible. Then that's where we want to focus right now. And I'm going to give you some ideas and actually example of one molecule why this is important, and in other aspects. I mean, and this is the part right here. I mean, what really is happening at the skin after a vector bite? And from here, I'm going to tell you why focusing on here is important, not only for understanding pathogen transmission, but also vaccine development. Okay. Now, if you go to any textbook in leishmaniasis, and that everybody knows that sandflies transmit, but the only thing that they tell you in most of the textbook is sandflies inject leishmania, and it goes from this extracellular to the intracellular form. That's it. What we have learned in the last few decades, and we're trying to put this in reviews, and we're going now in some of the textbooks, and I think this is very important because this is happening, is that insects not only inject the parasite, or mosquitoes, or other vector arthropods, they not only inject the pathogens, but also inject salivary proteins, and in the case of an infected sandfly, they inject also PSG parasite exosomes, and I'm going to discuss a little bit about insect microbiota. This is extremely important because we are completely ignoring this. We are assuming that the only effect is going to come from the parasite, but now we have to understand what's happening with this particular inoculum. And this is something that I want to stress, and if I will have to have a message to give today is this. The inoculum for vector-borne diseases, and as an example, sandflies, is very powerful. It's not only the parasite, it's coming with other components that are extremely important and extremely powerful. I'm going to show you right now. Then, just to give you an example, this is a few of the new from sandflies. We have sandflies that can inhibit the contact pathway of activation, very powerful molecules. We have actually a small protein of 11 kilodalton, which is a novel inhibitor of the classical pathway of complement. Very powerful, very strong. We have actually characterized in collaboration with Eric Calvo an endonuclease in the saliva of sandflies. This endonuclease destroys neutrophil traps. The parasites can go into neutrophils very fast. Can you imagine this is happening at the bite site in the first minutes? And we're isolated actually another anti-complement molecule from the alternative pathway of cascade, and actually it's also an anticoagulant. Then we have molecules that have anticoagulant properties and also anti-complement molecules, properties, dual properties being injected. Now, I want you to consider something. When we take medications for pain, for any other medication, we take like probably milligrams, or 500 milligrams of ibuprofen, just insects, saliva. The amount of saliva that is injected into the mammal, into us, is about, well, one pair of salivary glands is about between 500 nanograms to 1 microgram. And it injects probably 1/3 of that. And there are only like 30 molecules. Then these molecules are exerting their action with as low as 50 nanograms or 100 nanograms. That's how powerful these molecules are. This is something that I want to bring your attention. This is why we are studying this. To me, it's very fascinating. And actually, I have a kid now, he's 20-something. I started this many, many years ago. And my kid still asks me, Dad, are you studying still saliva? So, yeah. Why? And I start explaining. Now I think he's getting my point. But the thing is, to me, is the curiosity of how blood feeding are able to do this, to counteract the hemostatic system, has been incredible. But now learning that these molecules can affect parasite is also making it more interesting. Then I'm going to give you an example of one molecule that we haven't even published. This is we are learning about it. David Sachs showed before that when the sandfly bites an animal, it produces an influx of neutrophils. These red cells are neutrophils, and the blots are actually leishmania. And you see this with an infected sandflies. And actually, those neutrophils are very important because they capture the parasite, and then they take it to the macrophage. Then neutrophils are extremely important for leishmania survival. But guess what? You see it also in an infected sandfly bite. You also see lots of neutrophils. If you do needle, you just put needle damage, you see neutrophils, but probably 1/4 of this. Not only that, these neutrophils are sustained when you have an infected sand fly bite. Then we hypothesize that it's not Leishmania. Leishmania contributes because we know that there is a factor in Leishmania that brings neutrophils, but then we're seeing more or less similar activity with the uninfected sand fly. Then we hypothesize that there is a factor in saliva doing this, that is bringing neutrophils that are sustained. Well, I don't know how to do this. Let me see, yeah, right here. Oh, it went automatic. Then if you put just control, I don't know if you can see from here, these little cells, these are neutrophils coming from forming this gradient. And then you just put the HBSS or the other proteins and they go very slow, almost nothing. If you put saliva of any actually Lutzomia or Phlebotomos dubowski, you start seeing more, very fast actually, attraction. This is going to be very slow. And one of the things you see in the neutrophil is two phases. The neutrophils get like freeze, like they know something is going on, that gets polarized, and then it starts moving into the direction of the molecule. Then it may be working in two different mechanisms that we're still trying to study. Then one of the things that we do in the lab a lot is to characterize or identify the molecules that are doing this. This is whole saliva. We did a panel of 15 molecules, inject them actually as a plasmid in the back of the animal, and see which one bring neutrophils, and we found 2 molecules. And from there, we expressed the molecules, just to make the story short. And we did flow cytometry and TAC scan, and we showed that this, what is called 4C2 protein, one of them actually attracts neutrophils in vivo. Very small amount of protein attracts neutrophil in vivo. And if we do the in vitro assay, this recombinant protein also brings the neutrophils. Then now we have a protein. And this protein actually is the yellow-related protein. We're very surprised because this protein is also biogenic amine-binding protein. It's a very good marker of exposure, but also has this property of attracting neutrophils. Now, chemokines. This is a chemokine-like molecule. Chemokines have never been described in insects, actually in invertebrates. Then this will be the first arthropod molecule that has a chemokine-like function and does not have a chemokine structure. We have the structures, does not resemble chemokines, but apparently may bind these decoupled receptors in neutrophils or other proteins. Then that's another thing we're learning from insects. Novel molecules that may have novel drugs activities, but why? Why is so important in Leishmania? If you have animals together with Leishmania together with this protein, this molecule brings neutrophils, and there are more neutrophils infected with Leishmania versus control. And if we inject these animals with Leishmania, I don't have the graph here, there is more lesions of Leishmania than the control, suggesting that the Leishmania is taking advantage of this particular protein to infect neutrophils and to be protected. Then that's what the importance of these particular molecules in sandfly saliva. Then we just hypothesized that saliva is bringing these neutrophils to have this Trojan horse effect to go into the macrophage, survive, and cause the disease. Any question about at this point? Because I'm going to switch a little bit of the topic now. Are you sure?
Excuse me.
Any idea? Yes?
Is this the yellow protein that's related to the pigment in the
They call it yellow-related protein because it's related to the decarboxylase protein from pigments in Drosophila, but it's not the same protein. It belongs to the same family of protein, but does not bind tyrosine or any of the small component. It binds biogenic binding amines, bioamines. It binds epinephrine, norepinephrine, and actually, go back. This structure right here is like a big pocket. We call it cratagonist. It brings small components, even if it's a 40 kilodalton protein, but brings very components, very efficient, very high to small components. Sequestrate. And in addition to that, now we know that it brings neutrophils. The part of the molecule that brings the neutrophil, we do not know because it does not resemble any chemokine. But we know that it's doing. Then we're talking to experts in this field. All right. Then also, well, if you read the literature right now, and I think this is going to be also for Karina and others that are studying saliva, and Alvaro, we hear a lot about this saliva doing Th1, Th2. Okay? This is what you start reading. Think about it. And then one afternoon I was like, okay, this is Th1, Th2. But Th2 is usually adaptive. Then it's going to take a long time. Then what will be the benefit of a fast feeder to have a Th2? Or to the parasite at the first moment. It's gonna take a long time. Then I said, it may be something else to this Th1, Th2 for the initial event of infection. What we're finding is actually that is related to the innate immune response, more than Th2. This is shown actually by the work by Matthew Rogers. We're showing that too. We're showing that, now what we're showing actually is in this, that the Leishmania parasites are taking advantage of the innate immune response more than the whatever is claimed Th2 environment. I think the Th2 is too late. This innate immune response is so fast. It's taking minutes to hours. Then I showed you already with the saliva, with the neutrophils, it's taking minutes and hours. 30 minutes we're seeing the effect, 6 hours, 8 hours, 12 hours, parasite hits. Sharon already showed with the exosomes. And Sharon is going to come here in September and show you. I'm just going to give you a brief summary. It activates the it activates the inflammasome, producing IL-1 beta, and bringing, guess what? Neutrophils. Again, then the story, what we're seeing here is that these bites are targeting, or the pathogens are taking advantage of the innate immune response generated by the saliva to get established. We're not seeing much of a Th2 actually. Then I think we have to think about this. It makes more sense. I mean, at least to me. If somebody says I have another idea, well, I'll be happy to discuss that. But that's something that a conversation with other investigators. Anything here? Any other questions? Because I'm going to switch now to another topic. Then, the idea of saliva that promotes infection has been shown since 1988 by Jose Ribeiro. We have demonstrated this with the neutrophil story. We're going to, hopefully, Anderson will finish the paper. We're going to send it out. But that data that I show you, the neutrophil, we show exacerbation of the disease. But also, I just want to give you an anecdote about how we found this. This is work that I was doing this as a postdoc in Jose Ribeiro's lab, but I was collaborating with David Sachs and Jasmine Belcade. We wanted to reproduce this experiment, and then we were injecting the animals many times. We went to an animal facility, came back, and there was no infection. I said, oh no, we have to reproduce this. And the idea that's where we're going to put a lot of saliva. But actually what we were doing, we were creating immunity to saliva, and we were seeing protection. Then after that time, I stopped everything and said, you know what, I want to know how saliva produce protection and what are the molecules responsible for this effect. This is, after many years, this is more or less what I can tell you. There is no cross-reactivity between saliva and a pathogen. We haven't seen any cross-reactivity. Antibodies are not important. Antibodies, I can use B-cell knockout animals. They don't produce antibodies. They produce very strong immune response, and they protect. Then what we're learning is that either when we do an infected sandfly bites as a immunization, we see the same effect, or specific sandfly recombinant salivary proteins. If these proteins can produce CD4, can activate CD4 T cells to produce a Th1 profile, mainly interferon gamma, no IL-4. This is the only time that when you see the Th1, Th2 for me makes sense. Okay? Then I'm not saying that Th1, Th2 doesn't exist. For infection, I think the innate immune response is important. For protection, if you have Th2, there is no protection. We have seen that. If we see proteins, salivary proteins, and we have actually two, and one of them is the yellow, actually. One of the yellows. There is a T cell response, but there is Th2, there is no protection. We published that already. But if you see this Th1 profile in the rodent model with interferon gamma production, we see against cutaneous leishmaniasis in the mouse model and against B-cell leishmaniasis in hamsters. Very, very strong protection. Then the question was, can we do the same in large animals? And the answer is yes. We were able to reproduce this. This is work by Fabiano Oliveira in the laboratory. And this work took many years, about 5 years. We started in early 2010 and 11. And we demonstrated that uninfected sandfly bites protect monkeys against leishmaniasis, but we wanted to know the molecule. Then after what we call reverse antigen screening approach, we found one molecule, PDSP15, that produced a Th1 response in monkeys. We injected these animals and challenged them. This is very important what I'm going to say. Challenge them against infected sandfly bites. Most of the challenges against leishmaniasis in most groups are by needle. And David Sachs and we also demonstrate that if we do the challenge by needle, you see protection. But if you do by bites, the protection is gone. And you more or less know the answer why now, because of the infection inoculum. But how can we counteract the infection inoculum? And I think it's coming from this Th1 immune response. Actually, I'm going to tell you here. Then what we see in these rhesus macaques is that animals that were immunized and produced a strong immune response to SP15 were protected. The control didn't. Those monkeys that did not produce an immune response to SP15, cellular immune response, and were vaccinated, were not protected. Sometimes negative results actually help you a lot to understand the correlates of protection. Then this for us was incredible, was very good. Then the other aspect here is very important is, and actually that may help us to understand how this protein is working, is that animals that were vaccinated with SP15, and then you stimulate those, and protected, and then you stimulate those cells with Leishmania antigen. Remember I told you there is no cross-reaction. But in this case, what we're seeing is that animals that were vaccinated with SP15, and then you stimulate with Leishmania cells, PBMCs, you get a lot of CD4 T cells producing interferon gamma, indicating that was a very interesting result because this indicates that the Th1 produced by saliva accelerates immunity to Leishmania, meaning that probably these molecules are not like a true vaccine, but they may work in like an adjuvant-like molecule, presenting the antigen in the right context. Then this is the protein is actually novel protein of 15 kilodaltons. You don't see this protein in humans, only in sandflies. Then this is one of our leading candidates, and we're in collaboration with Peter Hotez. We just got a grant to do preclinical evaluation to hopefully move this to clinical trials. Then this is more or less how we think, or the hypothesis how we think the saliva works. When we immunize an animal, the antigen-presenting cells take this protein that present to CD4 T cells, and then the cells are primed against this salivary protein. Okay. Then, when you have an infection, then they have memory. They have memory against saliva. Then, when you have an infection, you have the parasite together with the saliva. Then, these antigen-presenting cells will take again the protein, but now they're going to show it to other cells, but also to these prime cells. And these prime cells are going to start releasing cytokines that are going to activate macrophage. And the Leishmania is inside of the macrophage. But also, something else happened here, that this macrophage that is being affected by the saliva immunity is actually, and these antigens are going to be presented to other T cells, but not now in Th1 context. If you don't have saliva, this is not going to happen. The parasite is going to be very happy in the macrophage, and it's not going to be shown. Then this is what we think. Saliva the immunity to saliva is helping to show the best antigens of Leishmania to later on have an immunity to the parasite. And that's what we saw in the monkey model, we're seeing in the mouse model, and we're seeing in dogs.
Given
Yes.
the importance of neutrophils in infection, in immunized animals, is any differential pattern of migration or differential attraction of neutrophils to the bite sites that you can tell?
I'm going to study that right now. No, I don't know. I do not know. Now that we are aware of the neutrophils, I'm going to talk to you. I'm going to show you something at the end related to that. But then in the terms of vaccine, and I think you're right. Now we're learning a lot about vaccines, but I think there is a big gap in that knowledge, which I'm going to discuss. Any questions about saliva? Because this is a very weird vaccine. Then we're calling it a non-classical adjuvant. This is a vaccine that is actually having an effect in a collateral damage, but it's actually very effective. Now, how do we translate this to the field? How do you translate this to the clinic? One of the things we wanted to know is like, can we produce an immune response, a Th1? Can humans produce a Th1 response? We show this in all these animal models. Then we did two, I'm going to show you this briefly. We did this a few years ago because I always ask the question at the NIH, like, okay, show me in humans. And if you're going to invest time in making a vaccine, you need to show me that humans can produce a Th1 immune response to insect bites, to the sandfly bites or to the salivary proteins. So, okay. Then we went into the villages of Kemen and Sogula in the district of Barauli in Mali, a few hours from the capital, Bamako. The only vector there is Phlebotomos duboski, and the only parasite is Leishmania major. We characterized that. But we wanted to know more about the humans. Could they produce an immune response? And this one, I can tell you the answer if you're interested, which is how long this immunity will last. I can give you the answer now. It lasts about 40 years, different to mosquitoes. In a cross-sectional study, we have different ages, but up to 40-something years, people can still produce an immune response to sandfly bites. Then, but to study the immune response, then we get individuals that we know that they are positive to sandfly bites. We expose them to 3 uninfected sandflies. Actually, these were duboski that was at the weed capture in Mali. We took them to the NIH, have a colony, make sure there was no parasites, not everything clean. We send the pupae back, the larvae back to Mali, and then they were becoming adults. And then we took them to the field and exposed humans. 3 uninfected sandflies, and then we selected individuals age above 18 years old, and then checked the DTH, and then checked for cell infiltration and cytokine expression. Then, I don't have the photo, but they produce a typical DTH response, textbook. But then we wanted to know if these individuals that produce a typical DTH response produce Th1 response, and actually the answer is yes. We see, at least with this marker of interferon gamma, and then we have the Th2 markers, the majority of these individuals produce a very clear Th1 immune response to the bites. Then we know that at least we can mount an immune response by the bite. Then I think the idea of moving this concept forward is good. But still, there is a lot of resistance. And if I ask you right now, will you believe in this vaccine? All of you will be like, oh my God, this is dangerous, it's going to give me an anaphylactic shock, it's going to be a problem. That's what we want to test, and that's the next point. Then, but also we want to know, one of the other bench to the clinic is to identify in humans actually what are the salivary proteins that produce a Th1. But then doing in the field was a bit complicated, although we decided to do it in more controlled settings. This was in collaboration with Walter Reed. We had a human protocol to expose individuals to uninfected sandfly bites, and we did actually 2 arms, one with Phlebotomos duboski and one with Lutzomyia longipalpis. The one with longipalpis is the one that I'm going to show you because it's the one that we could express more proteins. And the idea was this, is to not only understanding the immune response, but actually identify those salivary antigen that can produce a Th1 to move it forward for phase 1 clinical trials. And we just, Sharon Kamhawi created this device in plexiglass where we can have the flies, they breathe fine, there is this mesh. We can apply to humans for 10 minutes and many times, and then we follow the individuals and we take blood 6 months or 1 year later, get PBMCs, and stimulate them with different same fly proteins, recombinant proteins. Then we did this by a mammalian expression system. We use the same vector that we use for DNA vaccination. We transfect hex cells, purified by 2 steps HPLC purification, 1 nickel in the second molecular sieving. We get a very clean peak. The protein is more than 99% pure, but importantly, the endotoxin levels are less than 0.01 AU per ml in all this protein, except these 2 that we were not able to test because it was too high. If you use more than 0.03, you activate cells, then you have false positives. This is very important for immune assays that we're learning. Then just to make this story short, we tested many of these molecules, and 3 of these molecules, this, and this, produce Th1 immune response. These 2 actually from the same family. No, these 2 are from the same family. And we selected we cannot select everything, we selected 2, and this is called LGL143 or Lufaxin, and LGM19 or SALO, which is a salivary protein of anticomplement. You know, I have to tell you this. I have 2 sons. My first son's name is Isaac. And when I discovered the first anticomplement molecule, he was my oldest son, and it was in ticks. And I call it exotoscapulary anticomplement. Happy man. And then it comes a little one and said, Dad, and I couldn't find a molecule for my son. The second name son is Solomon. But then I found the anticomplement, and now it's a vaccine. That I switched the name is salivary anticomplement of Lutzomaya, SALO. Here it is. Then it has my son's name. He's happy now. Then I have 2 happy boys. But these 2 molecules are extremely important, and it's our leading vaccine candidate. We found them independently of other studies, and actually we showed that this actually has a very strong immune responses in dogs that produce Th1 response that lasts for 2 years in dogs. And this one protects hamsters against visceral leishmaniasis done by the FDA. Then we are in good track in terms of, this was now validated in another assays in animals. But we were very happy to see it in humans that are producing immune responses in humans. LGM19, the only problem with the LGM19 is only found in Lutzomaya, which is only found in these flies. But the LGL143, which is Lufaxin, which is the SP09, in Alvaro's lab will understand. SP09 is another molecule in Argentipes. It's found everywhere, actually. In all those flies around the world, you find LGL143, and it's very closely related. Then we may have a PAM vaccine. We may have a vaccine that may work for different flies if they're able to produce Th1 responses in these individuals. Then we're working in this Vaccine consortium to do the preclinical assay first, and we're done with that. And it's actually, this I have to clarify, we do not say that saliva is the vaccine. We are proposing that saliva is a component of an anti-Leishmania vaccine. We're proposing that saliva, because of the infection inoculum, then I'm going to be contradicting myself, because the infection inoculum is Leishmania, saliva, and other components, and saliva has been ignored, and Leishmania is always the target. I think the combination of various factors will give us the best vaccine. Then we're combining salivary vaccine, LGL143, with Leishmania antigens provided by Steve Reed and by Jaime Moreno in Spain, and combining a virosome particle that this was an idea from Edna Biotech in Italy, in Catania. And product development is being started with Baylor College, but then it was transferred to IDRI. We are done with the preclinical, but immunogenicity was published actually, and we're doing actually the toxicity in the Czech Republic, and hopefully, if everything is fine and we get all the approval, this is going to be done in Switzerland, the phase 1 human clinical trial for safety and immunogenicity. To me, this was the what can have the tipping point in saliva vaccine. I was like very anxious that, and I was like, oh, it's going to take me another 2 years to know if a vaccine is going to be working in humans or it can be done. Then, okay, done, I'm happy. I have to wait for this. I received a phone call from Matt Memoli, which is an investigator at the NIH that worked with flu. And he was contacted by the same team at, he's here in England, I think it's in London, SICK, the company SICK. They will contact him also that they have a mosquito saliva vaccine. And he contacted me, do you want to do it? I said, of course. Why do we want to do this? This is going to be actually the first trial of a vector saliva vaccine. And even if we say mosquito, I mean, mine, I wanted saliva of sandflies. But the proof of concept, the important thing, at least for this project to me, is to show that saliva is safe to use in human. Insect saliva is safe to use in human if you do the proper analysis and you take the proper molecules. Then what they did, they used Anopheles gambiae, saliva, peptides. They did animal testing, they show immunogenicity and protection. And the idea is that this saliva molecule vaccine is going to protect actually against Zika. That's the idea. I don't know if it's going to work, but again, remember, the most important thing is to see if it's immunogenic and if it is safe. More than immunogenicity is safety. Then it's randomized, double-blind. We have placebo control. This is in age is 18 to 50. There are 3 groups of 15. Only the saliva plus the adjuvant, Montadine, and placebo. And the vaccine is given subcutaneously. The hypothesis how this vaccine works is the same thing that I show you with Leishmania. Exactly the same principle. It's going to activate T cells, it's going to kill the virus indirectly, or to prevent the virus going into cells. They believe, I should not say the word believe, they hypothesize, because they have some data, that this vaccine also may work against malaria. They have data of about 60 to 70% protection in the animal model. I'd like to see this in other models, but they have some extremely good data. I'm not going to show you the data, because it's still ongoing. We finished the vaccination. We finished vaccinating. And the way we're going to test these individuals, actually it's going to be in our lab, is, well, we tested already, we exposed them to mosquito bites, just to make sure these people will not react badly to the vaccine. All of them were fine. Number one, safety. Some of the individuals complain against Montadine, against the, and this was actually happened before with flu vaccine. We also want to test if individuals that were vaccinated and then you put mosquito, if there is a mortality or development. And they showed this in the animal model, actually, that immunity to saliva may affect development of the mosquito, because some of the proteins may be important for blood feeding or for development of the inside of the gut. And the other part that we're going to test, actually we are developing the test right now, is to get PBMCs from these individuals, combined with Zika, activate cells and see if there's killing. But we want to start another study also for malaria. But at least right now our focus is Zika, and this is what we're going to be studying during the fall. We're going to collect because we need to collect the blood from all the individuals, do the assay, and then they're going to unblind before 12 Med. Then they may show the data. Then that's to me, that's very exciting, because something that started a decade or more ago about a concept in an animal model, now we're seeing moving into humans. We don't know up to this stage this is going to work, but I think this offers a completely new approach of combining, and this one also going to combine mosquito plus viruses later on. But this opened the door for a new way to vaccinate, a combination of something that for us was very difficult to think of, a combination of the vector and the parasite. And I think we're going to hear more in the next 2 years of this type of assays. It's going to be, there is a paper coming from Jessica Manning, that is one of the clinicians from this work, that wrote a review in a Journal of Infectious Diseases talking about this. Then it should come soon. It was accepted about 3 weeks ago. Then another aspect that I want to study yes, tell me.
So you're going to change the topic, right?
Yes.
Okay. So I'm thinking people that are constantly exposed to sandflies, for example, you would expect them to have some kind of immunity.
Very good question.
And people that get infected might be those that don't respond to that saliva exposure. So then if your vaccine is based on saliva only, I know you're combining, but I would expect perhaps that it doesn't work in those individuals that they actually need it. Those individuals that naturally wouldn't respond to saliva and therefore get infected anyway.
Very, very good question. And actually, I should have brought the slide. In Mali, another thing we did was ask that question. Okay, and well, we know that people can produce a Th1 response, some, because it was pre-selected because of the DTH response they produce. But if you do a study with, I think we put 200 individuals, and they take their PBMCs and stimulate with saliva, what type of immune response will they have across the villages? 25% Th1, 25% or 30% Th2, and the rest mixed. What is the proportion of naturally protected individuals in that village? It's about 30%. But we don't know if it's because immunity to the parasite or immunity to saliva. Now, the idea is to shift those Th2 or mixed responders to Th1. And we notice even in the animal models or the monkeys, if we do whole bites, there is a mixed response. And if there is Th1, if there is an individual molecule, we can shift that Th2 or mixed response to Th1. Or these individuals, or there's going to be a population that will not be able to work with only saliva. But we have a potential answer. I have to go back here. If this hypothesis holds true, that anti-saliva immunity drives anti-Leishmanian immunity, it means that if we find this either LGL143 or LGM19, imagine that I vaccinate, this group do not respond in the field to particular molecule. But if I'm able to immunize this individual with an adjuvant, LGM19, and the Leishmania, if this vaccination with the saliva protein and Leishmania, and then the second boost is, well, no. First vaccination is Leishmania, saliva only, and then you have a second vaccination with saliva with Leishmania antigen. The hypothesis is that this individual is going to have a strong Th1 response to Leishmania. Then in field condition, even if they don't respond very strongly to saliva, they will have immunity to Leishmania. That's the hypothesis. That's why I think we may have two ways that this protein may work. Yeah. And just to end, that I answered the question. It's tough. No, but we believe not all vaccines, even flu vaccines are not that good sometimes. Then just to finish here, I told you about because of what you asked, some individuals may not respond. And more importantly, most individuals in the field are exposed to mosquitoes or sandflies or the vector. When we do clinical trials, we try, at least in the US, is healthy human volunteers and probably were never exposed to sandflies or probably never to Anopheles gambiae, okay? But at least for malaria or for dengue or for sandflies, where are you going to use your vaccine? in endemic areas. And the endemic areas are going to have mostly people bitten. Then I was like, oh my God, there's another layer of complexity. And that's related to the immune responses and to the saliva and gut microbiota. Then now we need to study what's going on early on in humans. Then that's another bench-to-bench site that we just got approved. We were approved by the scientific committee, but we have to be approved by the, that's why it doesn't have an identifier. And we just want to know the early skin immune responses to icks and bites, and also to after exposure. And that's your answer, Alvaro. We want to know actually understanding early on through histology, system biology, cells, cytokines, chemokines, and how this will affect current vaccines. Any malaria vaccine, RTS, any dengue vaccine, any leishmania vaccine, how this is influencing our vaccines. If you think about it, you go to, we are aware, none of the vaccines against vector-borne diseases are very effective right now. Probably we're not understanding what's going on in field condition. And that's something we want to do now, just to get an idea, just to really know in humans, and we're going to move also to animal models. But this is just for me to and just to close. I think this is going to give us a lot of information, understanding the skin, the effects, the immune response, cells for vaccines, etc. Just want to acknowledge everyone from the lab and from Mali that helped us for all this study. And I didn't talk, sorry Alvaro, I didn't talk about the markers. I was supposed to, but decided to study mostly of this initial immune response. And with this, I'll finish. And thank you for your attention.
Okay, I think we have 5 minutes for questions. So I want to start asking. So if we don't think about the nature of the antigen itself, but as long as it activates a specific population of Th cells,
Yes.
you may have cross protection, looking into the magic, bullet that will maybe protect against several vector-borne pathogens.
Yes. Yeah, we think so. Well, a lot of immunologists right now are puzzled for this new type of cells, resident T cells or other cells that are coming very fast to the site, and specifically that they're like, oh, it makes sense. We see with saliva that if you're bitten in one site by fly, and then later on you get bitten again in this site, this reactivates. It's not only that the antigen is there, it's cell population that is acting fast is there. Then maybe this type of proteins, salivary proteins, are helping these cells to go fast and then present the antigen very fast or attack the antigen very fast. Then that's something that we are trying to learn. That's why all these innate immune cells are going to help us to understand how these antigens are processed or what they are activating. We know now the details. There has to be a Th1 immune response, but we have no idea how this protein can activate that. And many of them are without the help of an adjuvant. Then there is something going on there that we don't know. We have no idea. Questions? Don't be shy. Ideas? Will you take the vaccine?
What made you choose 3 bites for that initial study as opposed to something that would be more biologically relevant
Like one?
or over a season?
Oh, no. What we did that was what we call recall response. Then we did a Western blot and ELISA to detect individuals that we know that had immune response. What we wanted to do is to have a recall response. Then it's like the PPD test. If you're exposed to tuberculosis, then you do the PPD and you're going to have a 48-hour cellular immune response. Same thing here. We wanted to know if individuals will mount a cellular immune response to a sandfly bite. Then instead of using salivary gland homogenate, because we don't have the approval, human protocol approval, we use insects. Then, and we believe that 1 will be enough. But we decided to do 3 to make sure that at least 1 of them will fit. But usually the 3 fit very well. Yeah. In less than 5 minutes, they fit very well.
Okay.
All right.
Thank you very much and for this fantastic seminar.
Thank you.
And Jesus is going to be around. The whole of the anyone is interested, I can jump ahead.
Thank you.